Okay, well, good afternoon, everyone. And thank you so much for joining us this afternoon for our presentation in the ongoing 2021 CCOVI Lecture Series. My name's Sarah Ackles, and for those of you who don't know me, I'm the Marketing and Communications Officer at Brock University's Cool Climate, Enology, and Viticulture Institute, otherwise known as CCOVI. Today's presentation is going to last about 45 minutes, and then we'll have a question and answer period following that. That said, please feel free to type your questions at any time into the chat feature, and we'll get to them at the very end of the lecture, and we'll share them all with our speaker. That said, I'm very pleased to welcome our speaker here today, who's Dr. Wendy McFadden-Smith. Wendy earned her PhD in plant pathology from University of Saskatchewan in Saskatoon, and she's been working with growers to manage fruit pests ever since 1990. She's currently the tender fruit and grape IPM specialist with the Ministry of Agriculture, Food, and Rural Affairs, otherwise known as OMAFRA. She's also a professional affiliate here with us at CCOVI. On top of that, she works as an adjunct professor and sessional lecturer in the Biological Sciences Department here at Brock University. She's very active in extension education and is the author of the book Crop Protection Guides for Tender Fruit and Grape. Her current research projects includes integration of streptomycin alternatives for fire blight management, detection and management of spotted wing Drosophila, implementation of forecast models for fungal diseases of grapevine, and grapevine virus diseases and vector control. Today, she's going to focus mostly on the latter of all that I just mentioned today, giving us an update on her research on grapevine viral diseases. So Wendy, with that, I'll have you share your screen and you can go ahead and get started.
Thank you, Sarah. So, wait for that to come up. All right, I'd like to thank everybody at CCOVI for inviting me and apologize for canceling the last 3 years when I was supposed to be doing this presentation. So yeah, I'm going to talk about the work we've been doing with viruses in Ontario today. Wanted to first put my acknowledgements up so I don't forget them or run out of time. So we're very generously supported by OGWRI and CGCN, and part of our funding is through the Canadian Agricultural Partnership. Also like to acknowledge the grower cooperators. I don't have an experiment farm to work on, so I've been very lucky to have a good group of growers who allow me to do my thing on in their vineyards. Over the years, I've accumulated a large host of summer students who have been working with me and contributing to this. And a special acknowledgement to Rachel Ham, who is my research assistant, and Irina Perez, who was my research assistant. Some of the work has been done by the Pujari crew, Sud Pujari and his gang in the virus lab. Justin Rankema is part of the project. I'm not going to be presenting on his material very much other than just to mention it. And we had a lot of support from Jim Milwer's crew, Stephanie Bialik and Alex Gunn, doing some of the cold hardiness stuff. So we're going to jump right into it. Everybody's seen the symptoms of grapevine leafroll presented in presentations, so I'm not going to beat it to death. Usually we start to see symptoms in late August to early September, starting at the base of the shoot, and then it moves its way up. The leaves will curl downward, not upward, but downward, and the areas between the veins will turn red or yellow, depending on whether it's a red or a white variety. And the worst problem is that it delays maturation of the fruit. So this is from 2015, I believe, when we first started really recognizing how much of a problem these viral diseases are. So here's Leafroll with the on the left is an infected vine, and to the right is a healthy vine, not infected yet. So you can see the downward curling margins. Can you see my pointer, Sarah?
Yes, we can, Wendy. You're all good.
Okay. Okay. The downward curling leaves, the interveinal reddening. On whites, this is a shard. You can see the downward curling, very severe in shards, and the interveinal yellowing. And of course, the poor fruit development. You can see that this is an infected vine by the color of the leaves. And if there were a healthy vine next door to it, it would be much more advanced in maturation. Along came Red Bloch, recognized a while ago in the States, but more recently here. Symptoms once again appear August to early September. In this case, the leaves do turn red still, but there's no leaf rolling in either reds or whites. In the red vinifera, the symptoms tend to start out as kind of irregular pinky, crimson, red colors. It's a different color than what we see with Leafroll, but I think the way people see colors is variable. So anyway, it's not, it turns it red. The secondary and tertiary veins turn red, but that used to be a rule, but biologically the virus has not been very cooperative. So this is an inconsistent rule. So it's not something that occurs all the time. And occasional intervener reds between the secondary veins as well as the major veins. In white vinifera, the margins, the edges of the leaves turn yellow, and these will progress inward and progress to being brown, crispy margins. So on cabs, you can see that it's more of a reddish purple than a purpley purple in the reds on Cab Franc. Here's more on Cab Franc. This was back when in the day when we said that the veins did turn red, and it doesn't always happen. On Cab Sauv, once again, kind of reddish blotches, Pinot Noir. Symptomatic vines, delayed fruit maturation relative to asymptomatic healthy vines. This is Pinot Noir. In the whites, it's a little more discreet than the leaf roll virus in that the margins don't roll down. You don't have that. This is once again Chardonnay. You don't have that dramatic downward curling that you have with leaf roll. You will start to get chlorosis in between the veins. You can see here. And when it becomes more advanced, it actually starts to turn necrotic or dies. And this is where you get those crispy margins. And here's a more advanced one down here. The symptoms are variable depending on the variety. I forget where I got this from. It was a publication somewhere. So acknowledgements to wherever I got it, I apologize. Cab Sauv, Pinot Noir, Merlot, Chardonnay here, Chambourcin. Cab Franc, another Cab Franc, Pinot Noir, Pinot Gris, Chardonnay, rootstocks, pretty much asymptomatic, which is a concern. Another thing that's been observed, and this image is from a webinar that Patty Skinkas did with the Oregon State webinar, Red Bloch webinar series. And something that's been observed is that the maturation of the leaves is delayed in Red Bloch infected vines. So you can see here, this side is infected, this side is healthy. You can still see a lot of green. These pictures were taken at the same time, the same day. You can see that maturation in the Red Bloch healthy, the virus-free ones are it's advancing, whereas the infected ones, that maturation of the leaves tends to be delayed, and leaf drop tends to be later in a virus infected than a healthy. One of the tricky things about the about viruses in general is that expression varies from year to year. So this is another thing that really struck me in Patty's presentation. This is the same vineyard in 2008, and you can see all of the yellowing. You can see the reddening in the leaves. And this is the same vineyard at harvest in 2019. So symptom expression is very variable from one year to the next. And I think we noticed this even in 2020, where vineyards where we knew that there was virus, the fruit still managed to get enough sugar to be harvestable. And that had to do with the weather this past year. So the effects on vine physiology, I'm not a physiologist. I don't claim to be a physiologist, so I'm going to skip through this part fairly quickly. It's been seen that both viruses reduce photosynthesis. Red blotch also inhibits transpiration in the leaves. Reduce sugar movement out of the leaves. This one is characteristic of both. With red blotch, there's been a lot more work recently on the physiology, and Alex Levin did an excellent presentation on the physiology of red blotch, and I highly recommend that, and it's in the next slide. And what he's observed is increased starch, and I'm pretty sure the group in BC did the same thing. They found increased starch stored in the leaves, and this backlog of sugars and starches in the leaves triggers the maturation of the leaves. It's a signal for the leaves. And so in the reds, it will produce anthocyanins, and in the whites, chlorosis, yellowing. The other thing that they observed is that red blotch infected berries on red blotch infected vines are larger. So the concentration of sugar is lower, and this in turn has an effect on the secondary metabolism. So we're going to have fewer of those characteristics that we need in our finished berries and into our wine as a result of these physiological effects. And if you want a really good explanation of it, here's the webinar that Dr. Levin did back in October, and I highly recommend it. More in-depth explanation last week in the CGCN, last week or the week before, the CGCN webinar on viruses that was done by Sud and Pat Bowen. They discussed similar findings, less CO2, photosynthesis decreased, transpiration decreased, and photosynthesis was impacted even in green areas of leaves with red symptoms and in asymptomatic leaves, so vines that hadn't turned red yet, they were also having this effect. So these physiological effects are happening well before symptoms are developing. And once again, at the bottom is the link for that CGCN webinar if you want to check that out. We also were looking at it here in Ontario in a variety of, or in three vineyards, a Chardonnay, a Cab Franc, and a Vidal. And what you see in this graph are the pruning weights. So as a measure of vine vigor in the spring after, so in the spring of 2019, in the Chardonnay block where we had identified red blotch positive, Leafroll positive, and virus-free vines for both Cab Franc and Chardonnay, what you'll see is a significant decrease in vine vigor for both Leafroll and red blotch relative to the healthy ones. The effect in Cab Franc was a little less notable, but still, once again, reduced vine vigor measured by prunings. In 2019, 2020, we did the same thing, only we missed the pruning in the Chardonnay. So in 2019, we missed pruning in Vidal, and our vines got pruned. In 20, we missed the pruning in the Chardonnay, but some more interesting information. So here we have vines that are co-infected with both Leafroll and red blotch, with red blotch alone, with Leafroll alone, and vines that are healthy. And in this case, somehow, the vines that were healthy in the Vidal had lower pruning weights than the others, which is kind of counterintuitive. So we'll be looking at that again. And in the Cab Franc, we did see that decrease in vine vigor for both Leafroll and red blotch. I think one of the take-homes here is that the effects of the virus are going to be variable not just by year, but also by variety. Looking at some harvest parameters in 2020, this is in the Vidal block that was in the previous slide. The number of clusters per vine, the total weight of fruit per vine, the bricks, the average TA, the average pH. Even though this looks significant with the number of clusters on the dual infected versus the healthy, this did not shake out statistically. We couldn't find a difference among these. The only thing that was statistically different interestingly, was the yield on the healthy and the Vidal was lower than the other treatments. So go figure. The effects on bricks, relatively inconsequential, not significant. TA, pH, the same. So last year was a banner year for us as far as growing conditions. So this goes back to that image earlier on with the effect of environment playing a significant role. In Cab Franc, the number of clusters, pretty comparable with Red Bloch, Leafroll, and healthy. Total yield was about the same. The only thing that was significant in 2020 in this Cab Franc block was the bricks was a little bit lower, was significantly lower in the Red Bloch relative to the untreated. Once again, no difference in TA, no difference in pH. Looking at cold hardiness, some of this data I think I presented back in OFVC, not last winter. Wow, seems like a longer time than that. Solo Leafroll, so here we have the same color scheme where the dual infection is purple, Red Bloch is red, Leafroll is yellow, and healthy is green. And what you can see is that the healthy vines go into dormancy faster and more deeply by February than the infected vines, and Red Bloch, or the dually infected vines, seem to have the least cold hardiness overall. Similar effects in Chardonnay, 2019-2020. We're still collecting the 2021 data. So here, purple as the co-infection, and the solo infections in the healthy didn't really shake out. But once again, that co-infection seems to have more effect than just a solo infection. In 2018-19 in the Cab Franc, this is kind of odd. Well, we didn't have the earlier one, so we're getting the tail end. So you can see that the healthier vines were still, once again, more cold hardy, than the others, and the Red Bloch tended to decrease cold hardiness more than Leafroll in this particular case. And the same story again. So some of the questions about Leafroll and Red Bloch in Ontario: how common is it? What are the effects of infection on cold hardiness? I guess I just answered that. The effects of co-infection, I just answered that. And are there changes in distribution of infection over time? And is this relative to or relevant as far as vectors, and how do we control them? So looking at Leafroll and Red Bloch vineyards, these are vineyards overall in our survey in 2016, for which we sampled only Red Bloch, 2018 and 2019. The purple bars, the solid purple, represent red vinifera. The green bars, white vinifera. The striped purple bars are Baco, and the striped green bars are Vidal or white hybrids. And what I wanted to really look at here are these heights. And this is kind of the hidden danger, in that Baco and Vidal tend not to show symptoms as strongly as of infection as the viniferas do. So they can be infected, be a reservoir for inoculum, and not be recognized that they're doing that. So it's important to keep those in mind as well. Looking at incidence of Leafroll and Red Bloch between 2018 and 2019, once again, we're following the same color scheme. Purple is the co, red is Red Bloch, yellow is Leafroll, green is healthy. And 2019, the percentage of co-infections was higher, the percentage of Leafroll a little bit lower, solo Red Bloch a little bit lower, and healthy a little bit lower. Now this is not sampling the same vineyards. This is just a random sample across Niagara and Prince Edward County and Essex County. So we can't really say based on these graphs that there's an increase in disease. That seems to be what's happening, but we can't really draw that from here. But this part is kind of scary. Looking at just leaf roll, these are just going through and counting vineyards. Oh, sorry. And this was any kind of infection. So it could be 1 out of 20 panels sampled, or it could be 20 out of 20 panels sampled. So this is strictly incidence. It's not giving us a true measure of the severity of the infection, and that's where we go here, where we're looking at Leafroll virus, either alone or with Red Bloch virus. And what this figure is showing is the white with, I don't know if you can see the yellow speckles, is the percentage of the vineyards that were sampled that had less than 25% of those samples positive for leaf roll, and the solid yellow are those that were that had more than 25% positive for leaf roll. 2019, the same scale. We're seeing a lot more with more than 25% infected. Same kind of idea for Red Blotch, only red. 65% in 2018 had more than 25%. 2019, it was 54, but once again, that's a sampling thing. It's not the same vineyards that we're sampling. But this is pretty scary when you're looking at 50% of the panels that we sampled being more than 25%. And we'll come to where the 25% infection cutoff comes in a bit. So is there anything we can do to change the effect? We know they're here. What can we do to mitigate this problem? So it's interesting reading the literature and thinking that everything that you think is an original thought, and then you start reading the literature and you realize that everybody else has been thinking along the same lines. And so crop thinning we actually looked at for leaf roll virus back 2016, I believe, when Andréane did her undergrad thesis with me. And we looked at crop thinning, little to no effect. And this has been found for leaf roll and red blotch. Everyone that's looked at doing crop thinning has found little to no effect. Leaving it longer to ripen, no effect. Potassium fertilizer. There were some findings that the potassium levels in fruit that were infected with leaf roll had lower potassium, little to no effect. Irrigation, this had a variable effect, but only if there was a water deficit. So if you didn't have, if there was no water deficit, then the response to irrigation was minimal. And this is some of the resources for the work that was done in Oregon, looking at that. So this is where that information came from, some more resources for you. Here's a webinar where some of this information was presented by Cody Kopp, who's a grad student with Alex Levin, and another one by Patty Siskinkis that I presented earlier. And what's really interesting is Patty presented information for things in the Willamette Valley, and Cody presented from Southern Oregon, and some of the things they found were a little bit different, some of their observations. So that's once again an environmental issue. So definitely something to keep in mind when you're see virus symptoms one year and not the next, and also the response to these different stresses. I must have really liked that. I put it in twice. So another thing that occurred to us way back in Andréane's thesis and then to other people was abscisic acid. This is a hormone that's responsible for triggering the maturation of fruit. So I figured if the fruit weren't maturing, maybe we could push them to mature a little bit earlier by applying ABA. So Andréane tried it and didn't seem to work. We tried it again because there'd been also some work showing that ABA metabolism that leads to ABA was inhibited in infected vines with red blotch. So we tried this as a means to mitigate red blotch virus effects here. In 2 Cab Franc vineyards, 1 Riesling and 1 Chard, we actually identified vines that had solo infections of Red Bloch but not Leafroll, and healthy vines, applied Protone at 400 ppm once or twice starting at veraison, and then we harvested the fruit at commercial ripeness. And basically what we found was Red Bloch virus in the case of where are we? Chardonnay. Red blotched virus decreased the yield. ABA had no effect. And in the rest of the blocks that we looked at, red blotch decreased yield. In some cases, ABA tended to increase yield. Whether 1 or 2 applications was beneficial here, it actually looks like two applications wasn't as good as 1. Here, there was an increase in yield with 2 applications. So it's something that we're still working on, and I know Jim Wilworth is working on it as well. So we're hoping that that might bear some fruit, so to speak. Similarly, in 2020, that's the same graph. This is on Brix. So Red Bloch decreased Brix, no effect on ABA, no effect on yield. For Cab Franc here and Chardonnay. For Riesling, Red Bloch had no effect on Brix in either of these blocks. So there may be cultural differences or water availability differences among these blocks that could explain some of this. This is our work in Riesling in 2020. Once again, ABA didn't seem to make a difference. If you look at the total weight of fruit per plot in this Riesling block, Red Bloch didn't seem to make a lot of difference in 2020. Similar in the Brix and the TA and the pH. So 2020, optimum year, the fruit was able to ripen a little bit better. The Chardonnay block, on the other hand, there was a decrease in the number of clusters with Red Bloch, but the ABA didn't make a difference. ABA didn't make a difference here, and so on. So far, it hasn't done much for us. Okay, now I had a grower that was thinking outside the box and wondered, well, maybe mycorrhizae would help. So mycorrhizae are soil fungi that have a symbiotic relationship with grapevines. This is a figure that I pulled off the web that shows some of the different effects that mycorrhizae can have. And this particular grower was applying the mycorrhizae just to see if it would improve vine health at all. And we knew that there was leaf roll in this particular vineyard. So we did a trial where we compared with and without mycorrhizae, and the mycorrhizae that we used was BioCult, which is a Belgian product, registered in Canada, at 200 grams and 350 liters over 0.83 acres as a soil drench. So if you look at the untreated, so this is purple is Leafroll positive, green is Leafroll negative. So these are the virus-free versus the virus-infected. So not a lot of difference between the untreated and the mycorrhizal treatment. A little bit more difference actually in the Leafroll negative. So we had some response there, not statistical. And the rest, not a lot of difference. So not a big browsing success yet, but we're going to try and look at it again this year, I'm hoping. So a little bit more on viral transmission. Just to put your mind at ease, there is no mechanical transmission of either leaf roll or red blotch in vineyards. These are both phloem-located viruses, so they are not going to be transmitted from one vineyard to another by pruning, hedging, trimming, harvesting, suckering, cluster thinning, anything like that. And also there's no vine-to-vine transmission via root grafting. So the movement of the virus from one vine to the next by their roots joining together, doesn't happen. They are, however, vectored by insects. At least the Leafroll associated virus we know is vectored by insects. And the insects that will vector this virus are mealybugs and soft scales. For the mealybugs, the grape mealybug, obscure, long-tailed, and vine mealybug have all been identified as vectors of Leafroll virus. And the soft scales, Lacanium scale, and some of the cottony scales have also been identified as vectors. Mealybugs tend to be more efficient at transmitting Leafroll virus than soft scales. The early instars, so the ones first out of the eggs, are more efficient at transmitting, so picking up the virus and giving it to another healthy vine. The virus is semi-persistent, which means that it will pass. If a first instar crawler picks up the virus, it will pass to the second, the next growth stage, and the next growth stage, but not into eggs. Feeding by one mealybug crawler is enough to cause infection, and they can acquire Leafroll virus within 24 hours of feeding on infected vines. However, the symptoms of this transmission won't show up until at least the following year, if not later. So the first thing we did many years ago was determine which mealybug is present in Ontario. So back then we took representative samples, sent them off to a morphologist in Ottawa, and he identified them as grape mealybug. And once we got CCOVI in the show, we decided to have them tested by PCR. So once again, we have now molecular confirmation that the mealybug that we have is only grape mealybug. So what? Who cares? Sounds like an academic question, but there are really important considerations for this. And the first one is that grape mealybug has only 2 generations per year. There's the one that comes from the overwintering, and then one that happens in the summer. Other mealybug species can have 3 to 7 generations. So if you think each generation, the population increases in size, this is a significant thing. The second thing is grape mealybugs don't infest roots. So they will not move down from the trunk down to the roots the way other mealybugs do. That's important when you're thinking about management, because if the mealybug, for those other mealybugs that move down to the roots, when you pull out an infected vine, those roots remain in the soil, and those mealybugs could then feed on a new planting. The mealybug that we have does not move down to the roots. So when you pull out an infected vine, you pull out an infected vine and it's gone. The other consideration with which mealybug species we have is that pheromones are species specific, and this has implications for both monitoring and for mating disruption. So the grape mealybug, the first instar nymph, we've already talked about the fact that it's the most important, the best vector. It's also the most mobile. They will move when you peel bark. If you find first instars and come back in an hour, you won't be able to find a single one. They can walk from vine to vine. They can go up to 1 meter from one vine to an adjacent vine. They can be spread by wind. There's a study from 2008 looking at where they actually marked mealybugs and then had them release them, and what they found was that they could go up to about 8 meters distance. Some of the work in BC has shown movement and also elsewhere from one vineyard to another. And while the virus can't be transmitted by mechanical means, the mealybugs can be hitchhikers on machinery and even on workers. So it's important to know whether you have these very efficient vectors present when you're going out to do your vineyard operations. So looking at the life cycle, they overwinter as eggs and instars and first and second instars on the trunk. So this is a trunk where we peeled the bark back. All of this white is from mealybug eggs being laid. And here's a little video to show you what they look like. So each of these is a first instar, and this kind of webby looking stuff is the film that protects them underneath the trunk where the bark of the trunk where they're laid. So the mealybugs will progress from dark kind of salmon color through to a larger instar that is a little bit lighter in color, to larger and a little bit lighter, to full-size adults. So these will overwinter under the trunk, and then in the spring they will emerge up and they'll move onto the canes to feed. And where you typically find them is where canes have been bent to tie onto the wires. They like to get underneath that broken paraderm bark and hide underneath. So when we're looking for them, when we're monitoring, we actually peel the bark back on those canes and we'll find the mealybugs, and there's a little ant that's taking care of them too. So then that generation, so the overwintering generation is here. The females will move back under the bark and lay their eggs. We'll have the summer generation, and then the females will once again move back under the bark and lay their overwintering eggs for the next summer. So monitoring for grape mealybug, we use pheromone traps for grape berry moth, and in Oregon, this is an example where they are using pheromone ties or pheromones to monitor male grape mealybugs to time their sprays. We have tried these. We brought them here to Ontario. This one is full of midges. Here's one mealybug that we found, one male mealybug that we found in the whole time that we have been monitoring, and I'd say that we've been monitoring 4 or 5 years. We found one time, we found male mealybugs under the bark, and this past year, this is a magnifying loop. One of the students took a picture through the magnifying loop with their phone, and here's a male stuck on some of the bark. But so far, I have not been able to get pheromone traps to work to collect male mealybugs to help us with their timing. And that was why it was so important for me to identify that it truly is grape mealybug, because the pheromones for the other mealybug species wouldn't work. So I'm going to keep trying, see if we can get it to work. In the meantime, we're monitoring in 2 ways. We've put double-sided tape on the heads and on the canes of vines where there is significant mealybug activity, and this is the kind of thing we're looking for. Whoops, this is the kind of thing we're looking for. This is a mark made with a Sharpie, so you can tell how small. This is 0.4 millimeters from head to toe on this crawler. So the only way to really find them, I can't see them anyway, is to look with a dissecting microscope, and that's what my crew's been doing. The other thing is to actually peel back bark and look. This is very labor intensive, but here you can see a magnifying loop and a whole whack of mealybug crawlers. These are probably second instars on a piece of cane that the bark has been peeled back on. So why do we do this? So we know when we should be applying treatments. The products that we have available for mealybug management, oils, insecticidal soap, or the neonicotinoid clutch, we have for 2021, and then we won't have it anymore. It's going to be unregistered. These will only act by contact. So the mealybugs have to be out and exposed in order for them to work. The challenge is that we need to know when those crawlers are going to be out and exposed in order to know when we should be starting to apply these products. The other one that's a possibility that's labeled is malathion, and Ryan Brewster and I had a chat, and we were thinking, well, malathion has some vapor activity, so maybe it would have an effect on mealybugs that have crawled underneath that bark. So some of the growers gave it a try this past year. I'm not sure what the results are. We haven't managed to tabulate that. once again, the challenge is to figure out when we need to put these sprays on, because it's going to be early in the growing season, delayed dormant to probably just before bud break. So not going out too early, optimizing this spray, because there's only canes and trunk there. So we want to make sure that we're not wasting that product. So we're trying to work on finding a model to predict mealybug crawler emergence to help us with that timing, to at least know when to start the control. The other product we have is Movento. This one is biodirectional, that does move into the phloem tissue and then moves up and down the trunk. And the reason that is a good choice for us is that it will actually move from the leaves down into the trunk where the mealybugs are feeding or wherever they're hiding. The challenge is that the leaves don't export a lot of sugars before there's at least 5 leaves present. So in order for the leaves to take that sugar and move it down into the trunk, there's no point in spraying before around bloom. So the recommendation at this point is to apply the full label rate. The maximum amount per year is 920 mils. So my suggestion is to split that in 2 to 460, apply it twice, starting at post bloom and 30 days later. One thing I want you to keep in mind is be conscious of pollinators. Even though grapes are self-pollinated, the products that we apply on them can affect pollinators that are visiting cover crops or weeds that are in bloom within the vineyard, or even in areas outside the vineyard. So make sure to avoid spraying when those crops are in bloom, or at least mow them, and minimize off-target drift. There's the potential for biological control in some vineyards where softer chemistries are used. The natural control of these insects is pretty effective. The challenge with using introduced biocontrols is that most of the mealybug and scale are under the bark, so it's difficult for beneficials to find them. One of the strategies that occurred to me, and apparently to lots of other people, was ants are helping to maintain these mealybugs. So is there something we can do with the ants? And there is actually a project in the States, and there are products that are registered now to control the ants that are actually taking care of the mealybugs. They'll clean them, and they'll eliminate any competitors, and in return they get to eat some of the honeydew. It's very difficult to show efficacy for that, though, because you need really large plots and a lot of ants, and there are a lot of good ants out there too. How do you know if you have mealybugs or scales in your vineyard? Peel back some bark and on the trunk and look. This is like a massive infestation. You can see the mealybugs here. Sometimes if you peel off the bark, you can see them. This is an egg mass, first instar, second instar. Look for ants. If you have a lot of ants, start looking for mealybugs. If you have sooty mold on your grapes, mealybugs. Mealybugs will secrete a honeydew, and fungus that grows on the surface of grapevines can start to grow on that honeydew. So if you get to this point, you're pretty much the horse is out of the barn. But you can always start considering control for next year. Scales are the other thing that you need to watch out. In some vineyards, we'll see a lot more scales than we will see mealybugs. So these are adult scales. Some of these look like they've been parasitized, and these are some younger scales. And you can see the progression in size as they go from the younger stages to the adults. And here's a cottony scale, an ant taking care of it. This is where the eggs are in this cottony scale, so they can also transmit virus. Switching to Red Bloch, the big news was that the 3-cornered alfalfa treehopper was identified as a vector of Red Bloch in California. And then later the alfalfa three-quartered alfalfa, right. So here it is on a grape petiole. The leaf girdling causes reddening. So if you see this kind of red leaf, just odd red leaves in your vineyard, take a look and see if you can see the girdle around the petiole. The other thing that happened was in other parts of the country, buffalo treehopper was determined to also be a potential carrier of red blotch. And this also leaves that kind of girdling effect on shoots and petioles. And this one has been found in Ontario. So does red blotch spread? This is a block of where we're doing some of our research. This is Baco on this side. This is Vidal on this side. And there's a bush area here, an open field up here. And what we see, contrary to what we expected, is that the highest concentration of red blotch is at the end toward this in the Vidal, is in this at the end closest to this completely infected block of Baco. So the assumption is that it's moving from the Baco into the Vidal. So we have been looking for vectors there, and so far things are still up in the air. So Sid and his crew have been looking at doing some experiments with whether Red Bloch can be vectored. So they've used potted sentinel vines and placed them out in a very infected vineyard. And I'm going to let Sud talk about that more when he has his time. But we're working on it. Justin Renkema has also been looking at vectors of red blotch, looking at some of the work from the States. These are all species that are capable of vectoring red blotch, and these are species that we have found here in low numbers, but we have still found them. The bottom line with the vectoring is, so far there's a lot of money and time been spent, not just here, but in the States, and nobody has been able to categorically identify any major vectors of red blotch that are consistently a problem in the field. So it's still up in the air. It's possible that the vectors are there for a very short time, or they may be there only 1 year and not the next. It's really uncertain at this point. We don't know a lot about what is vectoring red blotch. Finally, propagation of infected symptomless vines is probably the #1 way that viruses are being transmitted. Looking at in 2020, we shifted from looking at mature vineyards to looking at newly planted vineyards. So we surveyed vines that were planted between 2018 and 2020, and 54% of all of the blocks that we sampled had no virus. That's the good news. The bad news, 20% had red blotch, 15% had leafroll, and 11% were infected with both red blotch and leafroll. So in the negatives, a whole range of varieties. In the positives, Cab Franc, Chard, Pinot Noir, Sovereign Coronation, and Marquette. So that's kind of a flag right there that table grapes and cold hardy grapes are not immune. In the leafroll, Cab Franc, Cab Sauv, Gamay, Riesling, and Vidal. And in the co-infection, Cab Franc, Gamay, and especially Baco. This was pretty scary. And looking at the percentages in the red blotch, anywhere from 1 to 18 out of 20 of those blocks were infected. Of those vines within a block were infected. 1 to 18 for leafroll, and there were more red blotch than leafroll incidents in the co-infections. So what do? Demand virus-free nursery material. Make sure that the scion and the rootstock have been tested for virus. And Sud did a presentation 2 weeks ago on the certification program. Highly recommend that you take a look at that. Other management, remove infected vines. Up to 25 to 30% infected, it's still economical to remove the infected vines. My suggestion, mark symptomatic vines during the growing season and use the 1+2 approach that Dr. Von Bell suggested in his Triggs lecture. Can't remember when that was. Time is a blur with COVID. With the 1+2, you would take the symptomatic vine and the vine on either side and remove both of them. Managing vine stress is important, so consider water, crop load, fertility. Remove infected blocks if there's 30% or more infected, and manage the insect vectors. Just a reminder, there are lots of things that can cause red leaves, so don't panic if you see red leaves. If you see symptomatic vines, first check the trunk and the cordons. Crown gall can cause red leaves. If the trunks are healthy, if the trunks aren't healthy, then train a new trunk. If they are healthy, next eliminate nutrients. Do a petiole analysis. Is there a nutrient deficiency? Then apply a fertilizer. If not, go to virus testing. If it's positive, rogue or remove. If it's negative, help. Give me a call. We'll come and I'll talk and look at it. That's it for now. And if you have any questions, I'd be happy to take them. Thanks for your attention.
Great. Thanks so much, Wendy. Appreciate all of your time today and giving us all that information. I have to tell you that Mealybug video is probably going to haunt my dreams tonight, so thank you very much for that. But in all seriousness, some great information there for everyone who tuned in. So I'll start off with the questions now. We have a couple in the queue for you. The first one is asked by Josh, and he wants to know if the blocks you sampled were selected for having high levels of infection.
No. For the initial surveys, everything was random. Sometimes growers would try to coerce my sampler into taking a sample from a block that was heavily infected, but this was totally blind as far as the 218 and 219. In the 220, I just put out the word to growers and said, I'm looking for vines that have been planted 2018 to 2020. Give me a shout and we'll come and look at them. So definitely not biased to high virus infection.
Okay, great. And I'll just let you know too, it looks like you're sharing the presentation still because it's me up on the screen instead of a split screen of you and I. If you could, yeah, perfect. There we go.
Thank you.
Okay, so the next question comes from Tristan, and Tristan asks, are there any blocks being studied where every vine is tested for virus transmission throughout the vineyard, or is it all done with composite sampling methods?
We have actually 3 sites that we're looking at every single vine, and this is the 3rd year that we've done it. So we are tracking to see whether there are changes in the infestation with time. And that Vidal block that I showed in near the end, that's one of the ones that we're monitoring to see if the red blotch is moving.
Okay, thanks. And I think you sort of addressed this at the end of your presentation, but I'll ask it anyway, and then you could just maybe elaborate a little bit on it for Diana. She asked if does no vine transmission from root graft mean that nurseries would only have to have certified clean scion and not rootstock? I know you mentioned obviously to advocate for both, but if maybe you want to address that and just kind of expand a little bit for her.
Absolutely not. You need to have clean rootstock because the virus can move from the rootstock into the scion. So you need clean both.
Okay, fair enough. Yeah, and to be fair, she did ask before you got to that in your presentation, yep. But just figured I'd ask anyway, just in case, and to hammer down on that point for everybody. Okay. So our friend Jose out in BC is returning the favor from last week, and he's got a question for you.
Only one?
Well, it's a multi-parter, so I guess it's kind
Okay.
of in line with what you asked last
Give
week.
me one part at a time.
Okay. So he's talking about pheromone traps. Says he hasn't had as much success using these in BC. So he's asking whether you think male or females are produced under Ontario or BC conditions. Is the first part of it.
Well, all the literature says that mealybugs are not parthenogenic, which means that the females can't just make more females without being mated. In culture, I've asked entomologists this, and everybody tells me no, that the females have to have males, that they can be very short-lived, but they say that you need to have males. I think we're on to something, Jose, and I think that's another whole project for us.
Okay. And then just kind of along those same lines, he also asks that same question about New York and whether they've been successful in using pheromone traps.
I talked to Greg Loeb, and he said that they haven't looked at pheromone traps, but they can grow it in culture, and they get lots of males in culture. So I don't think they're using pheromone traps in New York.
Okay, great. And that's all from Jose for now. We have another question that came in from Marlene related to Movento. She asked, if we rely on Movento, how long before we get resistance?
Good question. I think I forgot to mention on that slide with Movento, first of all, that once you get the populations down of mealybug, you don't have to spray it every year. Because some of the blocks that we've been monitoring, we're spraying Movento several years in a row. The populations went right down, and they didn't recover. We haven't seen a lot of rebound, so we don't have to rely on it year after year after year. Secondly, biologically, because there's so few generations, the probability of resistance developing is lower than something like Drosophila, which Sarah actually said pretty well in her intro, with some coaching. She really massacred it the first time. I agree. It's a risk, but it's the thing we have that works the best right now. There is a minor use project looking at another product that we might be able to use as well. But good point.
Awesome, thanks. Okay, the next question comes from Phil, and Phil's question is about the three-cornered alfalfa hopper and how it was confirmed as a vector in California. He wants to know if this is lab transmission or field transmission, and if that vector was confirmed here in Ontario as well.
I think it was just lab transmission or greenhouse transmission. It has been found here. I'm pretty sure that both Sud and Justin have found the alfalfa tree hopper here, but not in the numbers that you would expect if we're going to have if we have a big upswing in Red Bloch. You would think that there would be a lot more of them around. They are pretty big. So it has been found. We don't know, and even entomologists, like real entomologists, not me, not like I am, even real entomologists are having trouble nailing down what the actual vector is.
Okay, thanks. And I don't see any follow-up, so that seems to have addressed it. I think that looks like all of the questions that have come in so far. I'll give everyone another minute or so in case they have anything else to add. Just wanted to also point out, just because I thought it was important for any of our non-COVI peeps who are listening today, you mentioned the work of Andrienne, who's one of our PhD students at Bloch. Just for anybody who doesn't know who Andrienne is, I just wanted to share that and share the work that she's
Everybody
doing.
knows Andrienne.
I assume as much. Always like to give a plug to all of our students for the hard work they do to help all the work that you guys do. So.
Two of my undergrad thesis students are going to be working on this stuff. Nadia Skoropsky is going to be looking further at the effects of virus infection on secondary metabolites, and Lexi Kozel is going to be working on the Mealybug model.
Awesome. Another little plug for them too, so that's great to hear. Okay, and Ian put in a question while we were waiting involving global warming. So he wants to know how global warming and climate change are going to affect Red Bloch going forward. Any predictions you have?
I think that it's a double-edged sword. If we get rain, then warmer temperatures are going to help our grapes to mature more. But if it gets dry, then I think we're going to see an exacerbation of red blotch because water-stressed vines tend to show the symptoms a lot more. And that's not even addressing the biology of any potential vectors.
Okay. Thomas Openlander would like to know, are you sure on the alfalfa, not buffalo tree hopper? I don't know if that's enough for you to go on or if he needs to expand, but Sud actually commented in the thread
Thank
here.
you.
We found buffalo tree hopper,
Buffalo.
not alfalfa tree hopper.
Okay,
So.
sorry. Other way around. Thank you very much for the clarification. I'm not an entomologist, I told you.
Teamwork here on the thread today.
That's right.
Okay, Carly has a question for you. Are only male populations of grape mealybug used to determine control timing?
In the States where they have been able to use them, yes. Here we haven't been able to find them. They haven't been able to find them in BC, so we're relying only on the bark peeling and counting and the double-sided tapes.
Okay.
And there is commercial mating disruption for other mealybug species, but not for grape mealybug. So just be cautious when you read international journals or articles from elsewhere, because what works there may not work here, especially because of the vectors that we have.
Okay, very good. So it looks like everything. Is there anything you wanted to add, Wendy, before we finish up, just about some of the next steps on your research or what people can look forward to next, and then we'll call it a day?
No, I think that's enough. It's time for a glass of wine.
Okay, sounds great. Well, thanks everybody for all the questions. It was a great conversation today. You'll be able to find Dr. McFadden-Smith's presentation on our COVI website at brochu.ca/COVI. That way you can also find her email there as well if you had any follow-up questions or wanted to work with her on a project or had any other questions about your specific vineyard or things that you're seeing while you're out in the field. We don't have a lecture next week. That's just in an observance to Reading Week that we have here at Brock University. So we're going to be resuming on February the 24th, again at 3 p.m. here on Life Size. Dr. Tom Lowry, a research scientist at Agriculture
Real
and
entomologist.
Canada. He's going to join us on the 24th, and he's going to share some research with us at that time. So keep an eye out on our website for more information, and I hope you're able to join us on the 24th. Thanks again for your time, Dr. McFadden-Smith. We really appreciate it. Hope you have a great afternoon and enjoy that wine.
Take care. Bye-bye. I will. Ontario wine.
Absolutely. BQA. Take care.